Hello everyone, welcome back to another part of the previous video series which I am making from the class of molecular basis of inheritance, that is different types of RNA, their function and their structures. So I have already discussed about mRNA and rRNA, that is messenger RNA and ribosomal RNA. Only one RNA is left, that is tRNA or transfer RNA. Now what are the functions? Let us first recall about the two types of, especially the function of mRNA, that is actually carrying all the information for making the protein from the DNA from the nucleus to the cytoplasm. It is carrying all the information after copying all the information in it by the process of transcription. So that means the mRNA is carrying the codons through which it is carrying the informations which are stored in the DNA, and it is carrying it from nucleus to the cytoplasm. Then it will be attached with the ribosome with the help of ribosomal RNA, and then the protein synthesis will begin with the help of transfer RNA. What is the full form of tRNA? Full form of tRNA is transfer RNA. And as the name suggests, transfer RNA, transfer means it will transfer something, some molecule. Which thing it will be transfer? It will transfer amino acid residues from one place of cytoplasm to the other place. And that is why this is also known as adapter molecule. The tRNA is also considered as adapter molecule. What is the meaning of adapter molecule? What is the meaning of adapter? We all know that what is the function of adapter. When we want to transfer some data from some place to some other place, then we use the adapter and we just store all the information, all the data into the adapter. Then we used to carry that or transfer that data to some other place. Say for example from mobile to or from one computer to the laptop or something else like that. So similarly in this case within the cell also, the tRNA is acting like an adapter molecule. It is just carrying that amino acid into it, that is the data. Okay, the amino acid are the data which it is carrying in it, and it will just transfer that amino acid from one place of the cytoplasm to the another place. That is to which place? To the mRNA or the messenger RNA, which is waiting there in the with the ribosome to make the specific protein according to the instruction of the DNA. So that is why the name adapter molecule is given to the tRNA. tRNA is the smallest RNA. This is the smallest RNA among all the RNA which are found in the cell. So among all the three RNA, tRNA is the smallest RNA. That is why its sedimentation coefficient is quite less. It is like 4s. This is the sedimentation coefficient of tRNA. And when talking about the structure of the tRNA, R.W. Holey was the scientist who first introduced the clover leaf model of tRNA. Who gave the clover leaf structure of tRNA. According to him, or according to this model of tRNA, how the tRNA is looking like, just see here that I am drawing. In the 3' end, actually tRNA is not a single-stranded, a very simple structure like mRNA. Unlike that, it will form some loop and some stem-like structure. That is why it's also called a stem loop kind of design or stem loop kind of structure. Just see here that this is the 3' end, which is started with adenine, cytosine and cytosine, and another one nitrogenous base. These four are remaining free. There will be no attachment, no base pairing, nothing. And these three, ACC, these three are constant irrespective of any type of tRNA which are present in the cell. Whatever type of tRNA these are present, the ACC, that is the first, these three molecule will remain same as it is or constant. And these first four nucleotide bases or nitrogenous bases are free. And after that there are so many other nitrogenous bases, and then they can just form this kind of a structure, and right after that this can form this kind of a loop like design. Then it will be continued like this, and as you can see that these strands are parallel, these are running parallel to each other. That's why whatever nitrogenous bases are present here, they can form a very firm attachment with the help of hydrogen bonds with a complementary strand. So that's why within this stem-like structure, these are the stem-like structure, this is the loop. Within the stem-like structure, we can find the nitrogenous bond, hydrogen bond. So bond formation can occur only within the stem-like structure. Bond formation within stem. These are the stem-like structure, but within loop there will be no bond formation. No bond formation within the loop. This is the first information you have to keep in your mind. Then this will continue like this. It will form like this kind of a structure. Then once again it will form a loop. Then once again it will be continued like this. Then once again it will be like this. It will be like this. And then it will be indented here like with the 5' end. So here you can find that these are like this and the 5' end will be like this. This one, because this is going to remain free. So here the nitrogenous bases will be present, and as these are running parallel to each other, these stem-like structures, so whatever bases are present, they can form the bond between them. So they can form the base pairing or bond formation. Rather than saying bond formation, let me just tell you about the base pairing. This can be considered as base pairing, which is found in stem. Only the stem kind of structure is showing the base pairing procedure, but in rest of the loop there will be no base pairing. So we can find 3 loops and 4 stem-like structure over here. And another type of loop is here. This is known as variable loop. There are different names of different loops. Firstly, let us tell me about this. Let me just tell you about this one. This loop that is present near 3' end, that is actually known as acceptor arm. This arm is also known as acceptor arm. Why so? Why this arm is known as acceptor arm? Because the amino acid can attach to this arm. So let me just draw out here. Say for example, this is one amino acid. This will come and attach with this site. So amino acid can attach to this site. That's why this stem is also known as acceptor arm or acceptor stem. Okay, this is also known as acceptor stem. Acceptor arm or acceptor stem. So amino acid will attach with the 3' end with this constant region. A specific amino acid will come and attach. After that, this is about the acceptor arm. This part is known as acceptor arm. Okay, right after this, there is another type of group like this. Usual components are present, but as well as some unusual components are also present, like pseudo-uracil molecule is present. Then ribothyamine is present. T for ribothyamine. Then the T for ribothyamine molecule is present. Then 5 for your pseudo-uracil molecule is present. And C for cytosine is present. So this is present. That is why this loop is considered as T5C loop. This loop is considered as T5C loop, and this stem is considered as T5C stem, and altogether this is considered as T5C arm. So one is acceptor arm, number 2 is T5C arm, which is consisting of T5C stem and T5C loop. In the T5C loop, there is no base pairing. Base pairing is absolutely absent here. And after that, there is another stem, but before moving ahead into that, just see here that there is also another short loop kind of a structure is formed. Here actually the bases vary greatly. That is why this loop is known as variable loop. This loop is often considered as variable loop. Next, there comes the anticodon loop. Actually, this loop is known as anticodon loop. 7 to 8 nitrogenous bases are present in this loop, which are not forming any kind of base pairing. Among them, 3 bases are present here, which are known as anticodon. That is why this loop is considered as anticodon loop. So, I hope that you remember the structure of the mRNA, on which I said that with 3 nitrogenous bases, 1 codon is formed. 1 codon is necessary for a specific amino acid. So, here comes the exactly complementary bases. So, say for example, the mRNA strand which was present there, if that is carrying AUG, that was actually carrying AUG, which I draw. So, AUG, so what will be here present? Here will be UAC. So, if here the UAC molecule will be present, then this tRNA will carry methionine on it and it will go and attach to the mRNA. Okay. So, during the translation process, that is during protein synthesis process. That is why this loop name is known as anticodon loop and this stem is known as anticodon stem. The name of the stem is anticodon stem. And after that, this is the last, this is known as D loop, this is known as D stem, and all together these are known as DHU arm. DHU arm means, DHU means, you know, dihydro, there is one unusual component which is once again present, that is known as dihydrouridine. DHU for dihydrouridine. This is another unusual component which is present in the D loop and D stem, that is DHU arm. So, this is all about the clover leaf structure of tRNA. Now, in this structure, we can say that as the stems are running parallel to each other, that's why it is easier for the nitrogenous bases to form the base pairing among the complementary bases, and in the loop there is no base pairing occur. And in the loops, actually there are different types of loops according to their functions and everything. So basically you have to remember that in the 3' end the amino acid gets attached, and this is the anticodon loop which will actually recognize that where to get attached. On the mRNA there are the nitrogenous bases, there are the codons, on which codon I have to go and attach, and according to that it will carry the amino acid, because codon is very specific to the amino acid. As I said that AUG can code for methionine, so it will be recognized by the tRNA which has anticodon of UAC, and it will go and attach with the methionine amino acid. Then it will carry the methionine from one place to the AUG site of mRNA, and after that the transcription will begin, or initiation of the transcription process will start, and then the protein synthesis mechanism, translation process will start, and then the protein synthesis mechanism will be started. So this is mainly the function of the tRNA, that it will get attached and carry the amino acid from one place to the mRNA for the protein synthesis mechanism. And this is about the structure of the tRNA. I hope that the structure and the function of all the 3 RNAs are clear to you now, and we can move further towards the central dogma and the RNA world in my next lecture video. So keep on watching my videos, and if you found this video helpful, then please don't forget to like, share and subscribe to my channel, and comment down below if you have any queries. Thank you.